Good evening and welcome to Research Tuesday. We're in for a real treat tonight. Tonight we're going to be hearing about childhood neurodevelopmental disabilities, but I'd like to first acknowledge the Kaurna people, the original custodians of the Adelaide Plains and the land on which the university's campuses at North Terrace, the Wake campus and the Thebarton and Roseworthy. My name is Professor Andrew Zanatino. I'm the Associate Dean of the University's Faculty of Health and Medical Sciences, and it's a real privilege to be here tonight. And I want to thank you all for coming along for what is the fourth Research Tuesday for 2017. Tonight we're going to be looking at childhood neurodevelopmental disabilities such as epilepsy, autism, spectrum disorders and cerebral palsy, and the ongoing genetic and genomic research that's being undertaken here at the University of Adelaide. And we're also going to be informed of the prospect of the development of effective therapies for these types of developmental disorders. We'll also hear a little bit about the factors that are influencing patient access to genetic and genomic testing, and also the support that's associated with these disorders, and a little bit about personalised therapy to treat these disorders. So we're really privileged to have our speaker tonight, Professor Joseph Getz, who is the Professor of Human Genetics here at the University of Adelaide and at the Robinson Research Institute. It's astounding accomplishments of Joseph and his team that they've actually discovered more than 150 genes that are associated with neurodevelopmental disorders. And Joseph is the National Health and Medical Research Council Senior Principal Research Fellow. This is a very prominent a feature of prominent scientists in Australia. He's also the Channel 7 Children's Research Foundation Chair of Prevention of Childhood Disability. So following tonight's presentation, we're going to open the floor for questions. So I would really welcome questions at the end of the presentation. But first of all, I'd like to welcome Professor Joseph Getz to the stand. So thank you.
Thank you. Thank you. Thank you, Andrew, and thank you everybody else for coming here and investing your one and a half an hour to listen to what I have to say. I'm already a little bit nervous because I usually sit where you are, and I enjoy the presentations of the other speakers, so I know how important it is to communicate well and in a clear manner and really convey the message. So I'll try to do exactly that, even though Andrew put quite a bit of pressure on me by listing all the titles and everything else which is associated these days with our names. I'm a scientist. I really don't get to see these individuals. So please take it this way, that I really work with a lot of professionals, clinical geneticists, neurologists, pediatricians, who really provide that link to the individuals and these families living with these disabilities. So all my experience is somehow distant, but I guess we all have to maintain our focus on what we do and do it to our best of our knowledge. So I would like to start with a few dot points about what is known about Adelaide. When I came here 23 years ago, I didn't know very much about Adelaide. I was coming somewhere to Australia. I knew about Sydney and Darwin. I guess Sydney because it was a nice place, and Darwin because there were always storms. But I learned through different channels that Adelaide is really famous for many different things, and some of them are listed here. The reason why I came to Adelaide was single-mindedly human genetics and the excellence of the team at the time working here at the University of Adelaide and located at the Women's and Children's Hospital. So these are some of the people who really worked with me over many years, and mostly in the top row, and many of them, or all of them, are still around. And these are the next generation leaders who really made already imprint, and we hope going to carry the flag of the leadership, not national only, but international, in the field of genetics of neurodevelopmental disabilities. So just to put it in a few dot points and for your information, some of you may know, others may not. Here in Adelaide, at the Women's and Children's Hospital, Professor Grant Sutherland and his team discovered the most important and still the most frequent cause of familial intellectual disability, the Fragile X gene. This gene is not only important for early onset childhood disabilities, but also for late onset tremor and ataxia syndrome, and as well as premature ovarian failure. So it is still really the major player in this field, and it's routinely tested for. You may or may not know in '94 that same team, led by Professor Malley, identified a first gene for epilepsy. At that time, we called it epilepsy. Now we refer to it as epilepsies. Simply over time, we appreciated that this is not a 1 or 2 or 10 disorders, but is a group of tens of different types of epilepsies, which we put under one umbrella. We discovered the first gene for so-called non-syndromic intellectual disability, which is pretty much just learning and perhaps language problems back in '96. Adelaide team was also the only team in Australia to contribute to the human genome reference sequence, which was published and appeared in early 2001. We were also among the first ones to use the so-called next generation sequencing technology, which now it's hopefully will be coming to the offices of different medical practitioners, be able to sequence our genes in a timely and effective manner, because that's going to guide the interventions for our better. And more recently, with a group of Professor Alistair McLennan, we have perhaps reclassified cerebral palsies as at least partly genetic disorders. And our recent data show that probably 1 in 4 individuals living with cerebral palsy may have the explanation in their gene, or at least part of the explanation of their disability in genes. So in terms of neurodevelopmental disabilities, I'm referring to them, but it is really a group of disorders of the problem of the central nervous system during development. These are intellectual disabilities, autisms, epilepsies, movement disorders like cerebral palsies, language disorders, vision impairments. So we really started to refer to them as neurodevelopmental disabilities because the boundaries between the individual groupings of these disorders are slightly disappearing. We see many cases of cerebral autism or with seizure disorders, so they can be easily classified as epilepsies, intellectual disabilities, or movement disorders. Traditionally, we use these large families, and here I put one family, which we resolved only recently. We still have some of these large families. These are more than 140 individuals across different states in Australia, more than 40 different affected individuals. And these families were really resistant, not the individuals, but their genes to reveal the cause of the problem of their disability. And it was quite amazing how the technology changed the way how we do, and with all the know-how and the skills and, I guess, perseverance, we managed to resolve it just last year. That's more than 25 years of work of larger team of people. However, recently, we more and more start to study, because we have the technology, these singleton cases, where an affected child is born to unaffected parents. And really, it's good, these are the most frequent ones, and these are really the ones the system kind of ignored, because it was really hard to try to find the solutions to the cause of their disabilities. But with the new technology of whole exome, or so-called whole genome sequencing, where we, in a cost and time-affected manner, can interrogate pretty much the entire gene content of an individual, we can tackle these difficult cases. But we're still really playing on the knife edge, because we don't know whether that disability is due to genetic factor or is non-genetic. I'm a geneticist, but I don't believe everything is genetic. I'll declare it right at the beginning. So for many years, we really played and looked into the crystal ball to try to really find the answers for many of these families. And I have really put here the picture. I just wanted to share it with you at least this way as a ball of stem cells from one of our PhD students, which we are using just upstairs here and very soon in the new medical school on North Terrace. So just to remind you, human genome was sequenced or completed in 2001 and then cost a lot of money, and many people across the world have been involved. Now we can generate human genome sequence for less than $2,000, and by now we hoped it's going to cost $100, not $2,000, but thanks to the international monopoly and perhaps other complex reasons which we not necessarily understand, the prices are still fairly high. To be really accessible to everybody, because that's actually a very important point. I'll get to it towards the end of my talk. So we can generate these about $1,700, and then on top of it comes about $5,000 to actually make sense out of it, because just to have the book doesn't mean we read the book and we understand the book. We really need to look at it very carefully to find where the misprint actually happened and whether that misprint is actually relevant and does change the story of the book, because that's basically what we try to do. Now, there are resources. You can look at this reference human genome. You can explore it. There are now large databases where you can look at 60 or 140,000 individuals and their genetic variation in different genes. So these resources are building, and that's exactly what we need. It is a privilege to have the genetic information decoded, but I see it also as an obligation to share it with the others, because that's the only way how we really can progress faster and help those in need. And that's not just neurodevelopmental disorders, that's across many disorders: cardiovascular, late onset, Alzheimer's, Parkinson's. So if you look at, this is my professional lifetime. I actually finished university in '86. That's one of the first genes for rare disorders have been identified, and then it was going up. These are the milestones. PCR, that's polymerase chain reaction. For those who are not from the field, that's a magic trick rewarded by a Nobel Prize to generate a lot from nothing in no time in terms of DNA. Now, everybody dreams about this. Old alchemists wanted to do it. They want to generate gold. This is gold from nothing in human genomic terms. This is the opening or access to reference human genome sequence, but actually pieces of that sequence were accessible from about 98. So you see there was a spike. And then this next generation sequencing, again, that's a high throughput sequencing of everything for a reasonable cost, provided another spike. But we see that perhaps there is a decline and maybe we're reaching some saturation in terms of the number of genes implicated in different disorders and many rare disorders. So there's about 4,300 of them here, different ones. So when we look at some of the resources, there's not many. Over time, many started, but only few surviving to really cope with the load of information. Like online Mendelian inheritance of man lists over 3,700 unique genes, which explain nearly 6,000 monogenic disorders. So imagine to be in the office of a clinical geneticist, and you see a patient, and you need to perhaps postulate a hypothesis or speculate, is the disability of this patient genetic or it's not? And if it's genetic, where should I look? So there are a lot of guides. There's visual processing. A lot of these people are trained, of course. They have fantastic photographic memories, but still, the amount of information is overwhelming. So clearly the data processing, the international databases, and the aggregation of data not just genetic, but also the clinical information is crucial. Of course, not everything is as simple, that there is one gene and one clinical outcome if that specific gene is not working properly. That's what we thought. That's what Watson and Crick thought in 1953 when they discovered DNA. That's the central dogma of molecular biology, from DNA to RNA to protein to a phenotype. We already know that there is majority of genes that are associated with one phenotype, but we have a large number of genes where there is more than 1, 2, or 3 or 4 presentations. So in other words, that mean that you can have individuals with alterations to the same gene, to the same spot in the DNA of the gene, and they will present differently. Now, this is a complication, not just to diagnose them, but the value of genetics is a prognosis, because we have the crystal ball in genetics. We can look forward through the experience of other patients with the same disorder. If we diagnose here a 6-month-old baby, and we know that there is a family where there are 20, 40, 50-year-old in the United States, we just connect and we learn what's going to be the likely trajectory for the baby here. And that's what we want to achieve. And there is also experience where we can harvest potential therapies, even without knowing what is the cause or what is the molecular mechanism from gene to the disability. Of course, we have other complicating factor that many individuals would have more than one alteration in their DNA. Each one of us, on average, have about 1 to 2 completely novel, we call it de novo, alterations in, I would say, sensitive part of our DNA or our genome, which is called exons. So those are the bits which code for our proteins. About 1 to 2. And it's really just a bit of a game of nature and where these errors land. And based on that, we look different. Perhaps we have different sensitivities to different disorders. Some individuals are more fortunate than the others in terms of where these errors land. But each one of us, what is really kind of bad news that the more and older we get as parents, especially fathers, the number of these errors is going up, and quite significantly, which puts the question in forward about the age of when we become parents in view of neurodevelopmental disabilities, because there is a correlation between the age of the parents, in particularly father, and the risk of neurodevelopmental disability in the child. So looking at specific genes which we really work with, intellectual disabilities, if you go to some of these resources, 846 different genes. So that's probably a little bit more than the audience of in here tonight. So it's a lot of them. Epilepsy genes, autism spectrum disorder genes. I guess the autism is still a bit of a mess. There's only a few kind of high confidence genes, and there's a lot of genetic information associated with autism. So that's sometimes difficult to reconcile what it really means. For cerebral palsy, I guess the number is small, but we anticipate this to go up reasonably quickly, as well as schizophrenia, as an example. And there are very few genes which would cause everything, but there is at least one. This is a sodium channel gene. But there are many genes which overlap between different types of neurodevelopmental disabilities. Now, these are the networks which you can imagine if you're looking at the World Wide Web. It's really the types of problems which happen in these networks of these genes are really at the synapse. So that's when you connect your tool to the internet, whether Wi-Fi or heart. So that's where you make the connection. And that's how good the connection is. The communication depends on it. The size of the band, whether you go broadband or you have still the modem connection, or really you live somewhere in the East where you can't even watch a movie easily from the beginning to the end because the internet is just so slow. This is how some of the neurons actually look. So these are controlled. This is work from Lachlan, and I think he's here in the audience. Where you give this specific protein to the neuron, you see that suddenly from fairly usual one becomes very elaborate. So clearly these cells react to it. And this is usually a sign of connectivity, but also can be a sign of a problem in connectivity. But when we put the variant which we find in a patient into the same neuron, we don't see that response. And that's perhaps one of the tests where we can say, well, probably this variant renders that specific protein non-functional. And that's a very good sort of guide for us where to look, how to interpret, how to counsel the family, and how to perhaps start a swag of new experiments to see what's going on. Diagnostic rate overall in intellectual disability is about 50 to 70% of patients, especially those with more severe intellectual disabilities, are currently being diagnosed using next-generation sequencing. So that's perhaps is going to grow, but we also need to be appreciative that sooner or later we'll hit kind of a ceiling where we really need to look hard at whether the remaining cases are still genetic or they are non-genetic. And it's good to have these group identified because we can look for environmental causes, because they are much easier to manage and correct than genetic causes. Even though we have genome editing and even though we have stem cells, it's really not trivial to use any types of these interventions for the benefit of these individuals to correct their disabilities. So these are a list of few genes which I've been involved in more personally, and I'll tell you a little bit about this specific one, PCDH19. And this is on a single chromosome. That's where my history goes initially. So what I learned really that not all unique variants we all differ 1 every 300 base pairs in our 3 billion base pairs of our DNA. So we have a lot of differences. Many of these are quite frequent. A lot of them are unique. Not all de novo variants, it means they are not present in parents and they are present in a child, perhaps with a disability, cannot explain their disability. They can be just passengers, so we don't really know. In past, only a few years back, as soon as we saw a de novo variant not present in either parent, we almost assumed that that was the cause of the problem what we studied, and that's really changed. A bigger trouble is that not all functional variants, so not all variants like I showed you with the neurons which affect the neuron actually can be linked to the disease, because we actually don't know very well where is the normal and how much these cells can tolerate. But for the time being, we need to take some tests for granted that when we see an effect, that we assigned relevance to that specific variant. So here I would like to talk a little bit about the effect of sex, and in this case I mean the chromosomal sex, the presence of 2 X chromosomes, again the presence of 1 X chromosome, so differences between females and males, because they are, well, as you know, but perhaps you don't know it from the perspective I'm going to try to tell you. So in cerebral palsy, as an example, there is 57% males with cerebral palsy as a ratio with respect to the females, while 51% in Australian population. So there is excess of males. Traditionally, this was because males were more likely to be diagnosed because of the X chromosome inheritance and because of other factors. More recent studies suggest that actually female are the more robust chromosomal sex. They can tolerate genetic insult in terms of mutation more than the males. To illustrate this, you can inherit a disability predisposing DNA variant from unaffected mother to affected son. And this is quite frequent actually in autism. A lot of people studied this and they found arguments that this is indeed the case. So there is a protective factor in female genomes or their biology to protect them against these kind of genetic mutations. I'll come to it. Of course, one can say they're hormones. Yes, perhaps, but hormones don't explain everything in this case. So this specific disorder taught us a little bit about what might be behind that robustness or the resilience of female sex with respect to the genetic mutation. Because in this case, it goes the other way around. The males who actually don't have that protein functional at all are not affected, while the girls who have one normal X chromosome and one X chromosome without that functional protein or gene are the ones who are affected. So this is an X chromosome gene, and this disorder we studied and found the explanation in 2008. And the other group, our colleagues in France, studied it and came to the same conclusion, but on a different set of individuals a year later. So here for those, the circles are girls, the squares are the boys. When it's filled in, it means that's an individual affected with that specific disability which I'm talking about. The dot in the middle means that the person carries that specific variant. So here you can see that from this affected mum, she passed that X chromosome to her son. And of course, every daughter of a father will have his X chromosome because he has one X and one Y. If he passes the Y on, there will be all boys. All daughters inherit the X chromosome. So every single daughter of a male with that mutation will be affected. So this was a really intriguing disorder, which was pretty much the opposite of what we worked with for 20, 30 years, at least in my personal experience. So I would like to show you a short video so I can actually let the mum of one of these girls tell you about it.
When Violet McManus was born, her parents were thrilled. She was their first child, a perfect little girl who met all of her developmental milestones.
She was learning to pull up, and she was saying mama and dada. It was just really, really great.
She was just the best thing in our lives.
Trista Pandaleos felt the same way when her daughter Olivia was born.
I was very excited I was having a girl when we found out. We were beyond excited. Everything was fine with the pregnancy.
These children look very different, and their families may not have much in common. But before their first birthdays, one significant, life-changing similarity would emerge.
When she was 10 and a half months, she had her first seizure. We flipped on the light and she was completely blue, and there was bubbles coming out of her mouth. So then we called 911, and she had hundreds of seizures within 2 weeks of being hospitalized. She was never the same child after that. She stopped speaking. She didn't know who we were. She was gone. I couldn't really formulate what was happening. I'd never seen a seizure before in my life, and I just was panicking. So I called 911 and just said, please come quick. I don't know if she's gonna die.
Over the years, doctors would try dozens of medications to control the girl's seizures, and their desperate parents agonized over what might have caused their child's illness.
So that's where I decided to stop it. The video goes for a few extra minutes. But the bottom line, I hope you understood. These little girls are born perfectly healthy and happy. And at about the age, I'll show you, maybe 8 months on average, they get their first seizures and they really regress significantly in all aspects of their development. I work with these ladies. They are very passionate drivers of the alliance in the United States, and there are alliances across the world which try to raise funds to sort of resolve this enigmatic disorder. So this is like a diagram of number of different mutations. So this is the gene, here is a protein. This is a protein which sticks out of the neuron and normally makes the neurons to bundle together. So if the neuron has this molecule, the other neuron has this molecule, they know they have to go together. So it's an identification mark for the neurons when they move early during development through the brain, because neurons do move as we develop. So when Christy and Ewan looked at how variable this disorder is, we pretty much see everywhere the loss of function of this molecule. But the outcome is really highly variable. 80 to 95% have these clusters of seizures, and that's why we call this disorder girls clustering epilepsy, to really put it into the head of a lot of clinicians that they have to look at it from that perspective in the first instance. Cognitive decline, frequent. Autism, pure autism, but autism with other neurobehavioral problems is frequent. But there are some which just don't have the problem. They're fine. And that's really intriguing. It's provocative. It's really disturbing. You would say, well, this is not the cause of the problem. Well, Christy has put together this table with 25 individuals published by different people around the world. These are all girls. This is a month's time when their seizures started. So until then, they were normal girls, as you heard from Julie and Krista. These are all sorts of different types of seizures. And this is the outcome from really normal to very severe. Now, what I didn't tell you is that all these 25 girls, they are unrelated. A lot of their mutations are de novo, so it means they happen. The parents don't have it. So the mutation happened in the same place. Out of 3 billion places in our DNA, it happens exactly in the same place. And this is the annotation of that place. So all these girls, what they have in common, they have exactly the same genetic alteration, yet they are so different. So why? Clearly this intrigued us because we can just give up. It's too hard. But we see it also as an opportunity to really understand why some of these normal ones are actually protected. Is there a clue for us to find a treatment? So we thought that the X inactivation, so this is the process when if female have 2 X chromosome, early during development, when there is a bundle of cells, and not long after conception, these cells need to decide to shut down one of the X chromosomes in female embryo because the life is not compatible to have 2 X chromosomes working in the same cell. So these cells kind of decide it's this chromosome or that chromosome. They usually do it randomly. So that's why females are all mosaics. You have 2 different types of cells. While we as males are only 1 type of cells, if I look at it really from the black and white perspective. So we thought that that was an explanation. Now it's interesting, and again, I had to learn this. That is not something I was taught at school, because at the university, I was actually out of university by then. But Ursula Mittwoch noticed that there is different developmental sort of speed for males and females. That the girls are usually smaller because they are about 1 week behind the boys at term. Now, this was only observed. Nobody knew the explanation for it. But the French team in Paris figured out that this is all because these female embryos have to decide which chromosome they're going to inactivate, and it takes time and effort, and that slows them down. And that really delay at the beginning multiplies over the time to about 1 week. And that's why the developmental stage of the female is slightly different at specific time than the developmental stage of the males. So we believe that this X inactivation, which is happening, might be friend or a foe. And that's what we studied. So with Paul Thomas here at the university and our joint PhD student, Dan Paderick, we used mouse. I'm not going to go into great detail, but this is a section through an early brain development of the mice at the embryonic day 14.5. And this is the cortex. And you see that red and green pattern. So here, using very clever engineering, maybe one of the first in the world, Paul and Dan labeled the good cells with the good protocadherin 19 and the cells with the bad one. So we can see actually what happens to these cells. And you can see they come to cluster together. We also don't see any yellow color, which would suggest that same cell would express both, because then overlap between green and red would be normally yellow. So you have this stripy pattern. Well, that's good. We know that the cells migrate from here up and they go in these streams. We know that they bundle together because that's the role of the protocadherin. So what's about it? Well, the about it came when Paul and Dan compared females which are identical, like identical twins, and look at their brains, and they are different. You can appreciate it, that there is more red and here is more green. So these are like monozygotic identical twins, which are different. And we have identical monozygotic twins with this disorder, which are discordant. One has epilepsy, the other one doesn't. So what they also did, they they look at these so-called spike wave discharges, which is a EEG measure of seizure activity of the brain of these mice. The wild-type mice, so those without mutations are fine. Those which don't have any of this protein on either chromosome are fine too, but only those which are the mosaics, so the stripy pattern ones, are the ones who have very much increased activity in their brains. However, there are some which are kind of in the band looking like normal. So those are the ones where probably the X inactivation didn't really tip the balance one way or the other. So in this case, the X inactivation is a friend. However, as soon as the X inactivation is really 50% as it normally would be, then it becomes a foe. That's where we see the biggest problems. So that's illustrated this way. If we see that kind of pattern, then we get perhaps these outcomes. If we see either bad ones or good ones, we tend to end up either unaffected or very mild consequences. So now it's complex enough, and now we really, by these families and by Julie and Francesca in Italy, we're really pushed, please, just can we do something about it? So we really put our frame of mind that way, so we could do something. So you here in the audience found something really unusual, and that is that this molecule, even though it works in neurons, makes apparently neurons to come together, regulates estrogen receptor. Now, many of you would have heard about estrogen receptor in association to breast cancer, all sorts of other problems, aging. Even more recently, dysregulation of estrogen receptor has been described in autism because these receptors are very active in the brain, and they regulate pretty much what they do. These are molecules which regulate other genes and tell them when to jump up and when not. So they are really the directors of the orchestra in those specific cells. And we found that many genes are dysregulated, and here I just mentioned one, not going to dwell on it, oxytocin receptor. You would have heard about oxytocin and autism and generally sociability. So this system is also dysregulated. So that was very exciting. Now what we also found, here it's really the lifetime of early, from infancy to toddler childhood. Here we see the levels of the hormones. This is the mini puberty, which lasts about 6 months. That's where we protected after we come to the world by hormones to actually face the new environment from intrauterine to outside. And this is a puberty when around 10, 12, the hormones go up again. Interestingly, when we cumulatively look at the onset of the seizures of these girls, it happens exactly about the time when they lose protection from the hormones and bang, then they get the seizures. And what is interesting for those who are old enough, but not all of them are old enough, they usually stop having seizures when they come to puberty. Many of them, not all of them. Again, perhaps explaining that maybe the hormones actually play some protective role. We know that estrogen is protective. And that's why hormone replacement therapy for older women. So what is interesting is that also, and I'm not going to dwell on it, I just mention it because some people may ask perhaps, this is the time when we get our children immunized. And it has been found and noticed for other disorders, these so-called vaccine encephalopathy. You heard about this, and autism and everything else. So this is the time when you normally get the onset of these seizures, and this is the time when you go to the doctor to get your child immunized. So of course, there are people and there are situations where it by coincidence happens. Now, whether there is more to it, or it is really just a coincidence that the vaccination, immunization unmasks the genetic predisposition, that's a very good question. But maybe there is more to it. Maybe there is something. Because what is also important, which I didn't tell you very much about it, if we delay the onset of the first seizure, we really give these girls a much better future. So later they start seizing, the better outcome generally they have in terms of their intellectual development, their social interactions, and generally behaviors. So my colleagues actually in Melbourne were the ones to show that for a different type of this vaccine encephalopathy, it was really just a predisposing DNA variant which was unmasked by the immunization. And SCN1A is actually the most important epilepsy gene. And interestingly and intriguingly, the clinical overlap of Dravet syndrome is very much similar to the clinical presentation of the PCDH19 girls clustering epilepsy. So there is more than that similarity, but I can't tell you much more. What we also found initially, and that's Stanley's work, that we found these neurosteroids were low in these girls, especially allopregnanolone. I don't know, maybe some of you heard about it, but when I started to look at it, I really nearly fell off the chair. Allopregnanolone has been used to treat acute epilepsy, which is called status epilepticus. Allopregnanolone treated mouse model of Fragile X syndrome, and it's in clinical trial of Fragile X syndrome. I told you, the most frequent intellectual disability, familial, gene found here among 3 other places worldwide. It is a therapy for Alzheimer's. It's being tested in post-traumatic stress disorder, and it is effective in postpartum depression. So these are really the hallmarks of the disorder. I tell you about girls clustering epilepsy from different perspectives because we study it on girls. But when they grow up and they become old, they have all these problems. They have behavioral problems, they have depression. So perhaps there was something in allopregnanolone. So we completed a study we just published on another set of these patients. And we found that yes, these molecules are downregulated, plus others, including cortisol, which is an important marker of a stress response. So maybe these girls really respond to stress very differently. And now I come back to the immunization, and maybe there is a bit of a stressor. There might be minor infection during that time, which precipitates perhaps. So even in the meantime, we can't treat, but perhaps we try to avoid the exposure to the stressors. Maybe that may have some help. However, we went further and we convinced at least this company to complete a clinical trial with the analog of these neurosteroids, which is called ganaxolone. And that's been completed. There's some good news in it, but not all good. All news is good. That's all I can tell you because that's being just prepared for publication at the moment. Is classified, Americans say. Other facets I wanted to talk about is neurodevelopment and cancer. Many of you probably would have heard about it. Down syndrome is a good example where there is high risk, for example, of different types of blood cancers. Among other things, they're also at a high risk of other comorbidities. This is one of the first genes I actually worked on back in France in early 90s. And we implicated it in this disability syndrome, but then it was implicated in other disability syndromes, and then a swag of different blood cancers. A number of genes like this started really to grow. When I looked at this paper, when they looked at different mutations in cancers, in pediatric cancers, and they look at the most frequently mutated genes, pretty much all of them apart from 2 or 3 are neurodevelopmentally important genes. So mutations in these genes, when inherited or are congenital, they cause neurodevelopmental disorder. But of course, a lot of these mutations, or virtually all, are somatic. It means that they happen at later stage of development. People start noticing risks between autism and cancer, and some people can attach even value to it. So these individuals are at higher risk of cancer. Now, it's really hard to say what these things have in common, neurodevelopment and cancer, and why. I guess that's still subject of a study. But to my perhaps naive thinking, it looks like that when we lose the function of a specific protein very early, really at the few cell stages, or really from the very beginning, perhaps we have a buffer, we have a capacity, like I talk differences between women and men, to find alternative solutions. So there is some rescue. However, if a loss of function happens abruptly in a cell which is highly differentiated, really working very specific tasks, and needs everything what it has to deal with the task, and suddenly you remove one of the components, then the cell can maladapt. And maladaptation is survival, cancer is survival. These cells survive beyond the control of the other cells, so they create a problem. So last bit, I guess, or one of the last bits, I would like to talk a little bit about the implications, financial and equality of access to genomic testing. Not that this is my favorite topic, but we really do need to talk about it, even after having NDIS, how much the care and good quality care costs for these individuals. And our colleagues in Sydney estimated this some time ago, that it's nearly $14.5, nearly $15 billion per annum. And most of this cost is really paid by the families with these individuals. So there is relatively little support from the government. What is really striking, and you may not believe me, that the cost of intellectual disabilities is more than cancer, cardiovascular, and stroke. I'm talking big diseases. Now, why? To me, because intellectual disability is lifelong. A lot of these individuals are born, or within the first year, they develop and they live 50, 60, 70 years, and they require quite intensive care. That's why they cost. It's also important to mention this, that when we use these genomic technologies, I didn't talk about it very much in a clinic, if we compare the cost of the genomic testing versus traditional standard care, we see a tremendous difference in cost, 27,000 versus 5,000. But there is a different aspect or different dimension to it, and that is also the time. Because this is one test with a follow-up. This is many tests across time, which also need to be reconciled by many different people. So there's really a concerted effort in Australia to make this testing accessible to as many people as possible, if not to all of us, through our medical system. So in 2015, we were awarded this large 25 million grant from the National Health and Medical Research Council, the Commonwealth agency, which is the main funder of the medical research in Australia, to look into the implementation of genomics into health. This is the last slide on that. It's very complex. It's only in the second year. But it's not only about doing the test. It's really looking at the diagnosis, stock take, how things are done, so we are really on the same page. And that we also support research, because these patients from diagnosis, they're not going to survive better. We need to manage them better. We need to implement personalized medicine, and this is the beginning of it. So it's also data management and sharing. Without sharing, there is not going to be benefit. And that's, I guess, all of us need to appreciate and look at the benefits or the risks associated with that. Economics, as I mentioned to you, and policy. And as of course, we need to educate our workforce. So these are huge opportunities for future generations in this space to really find their place. So in wrapping up, I have only a few take-home messages for you, not very complicated. And I just put them here simply. As you could see, I could convince you the architecture of these disabilities is complex. There's probably 1,000 plus different genes, and it's really hard to find the answer for a specific patient, but we are getting good at it. But it takes, probably a few years ago, it took up 1 year. Now maybe we can boil it down to about 3 months. It depends really how lucky we are and what type of change we encounter in that individual. There are still many for whom we don't have answers, and large families we don't have answers. And of course, those changes are hidden, or we really need to do more research to understand more about our DNA, how it's packed, how it presents itself. There are differences between men and women, and not only because of the hormones, but the X chromosome itself. And the fact that women and females are mosaics gives them a bit of an advantage, and deservedly, I should say. We see these co-occurrence, or we call it comorbidities, it's a lot of other disorders. So it's really the question, so what do we treat? Actually, for those girls, you saw Julie talking about her daughter, they are perhaps not that concerned about the seizures because they know that they come and go. They are really concerned about the behavior because they get tantrums, they don't look into the faces. That's their main concern. They really would prefer perhaps to have the treatments for the malbehavior rather than for the seizures. So that's also where we need to. We look at it as a seizure disorder, all drug companies will test, and if it decreases the seizure frequency by 50%, that's a drug goes on the market. Perfect. It is really, we need to look at these patients more holistically and listen to these families and individuals. They often can't stand up for themselves like individuals for other disorders. That's also an issue. We always have to get the information through their carers or through some who can actually comment on it. The early diagnosis, it's the beginning of everything, and we know it, and the recurrence rates can be decreased. The family is empowered, the knowledge and the connection, these people come together, they share the information that settles the guilt and move everybody forwards, really looking towards positive solutions for these individuals rather than dwelling and going around and around about the causes, what may have caused the problem. And at the end, I really would like to appeal to all of you that these disabilities, and I hope I don't need to convince all of you, really need as much compassion and support as cancer. We can see Kylie Minogue or other celebrities talking about their cancer experience. We don't see celebrities with neurodevelopmental disorders talking about their experience. We almost believe that that's not genetic, that's a mistake of the parents, that's a mistake of the system. We probably prefer not to look at them. I really would like to see, I see it from the funding perspective, these are really individuals and families which deserve most support and compassion because the causes of their problems are pretty much the same, maybe even the same genes as in cancer. So I would like you to take that message and perhaps spread the word to look at these groups of disorders at least equally. This is the team, at least part of the team. These are my senior colleagues. Students like to have fun, and that's important. That was one of our Olympiad at the end of 1 year. This science cannot be done in a small laboratory. It's an international science, multidisciplinary. I don't have any single author publication, maybe back from 1987, I think, when I was really still young and arrogant. And I didn't appreciate the support of other people around me. I submitted a paper only with my name. These days, there are really teams of 40, 50 people who contribute with different expertise, clinical, cellular, molecular, stem cells, genetic, you name it. It's really very important to have these people working together. And there's no other way. Because for many of these genes which are listed to you, there are probably about 5 patients around the world. We're not talking thousands of individuals with mutations in a lot of these genes. There are about 5 or 6. That's usually enough to implicate them as a gene important in that disorder. And then all diagnostic laboratories, testing laboratories around the world can take advantage of it, and then the numbers go up, and we better understand the clinical presentations. We're refining, go back and forth, until we understand what's going on and perhaps can start thinking about interventions. And these are our funders. Without them, I wouldn't be here. I'm very grateful that I am here and acknowledging their contribution. So thank you very much for your attention. I would be happy to answer questions if you have any. Thank you.